Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor and Head of the Department of Biochemistry, St. Joseph's College for Women. Today we'll be discussing the topic glycolysis and its regulation. The topics that will be covered in this video are the salient features of glycolysis, the various reactions of glycolysis, then production of ATP in glycolysis, followed by glycolysis and the shuttle pathways, and then regulation of glycolysis. So what is glycolysis? The term glycolysis, it's actually derived from the Greek word glykos, which means sugar, and we know that lysis means dissolution or breakdown. So the glycolysis, it is actually a universal pathway which takes place in all the living cells. And this pathway, it has been thoroughly elucidated and chalked out in the year 1940. And it is referred to as the Embden-Meyerhof pathway or EM pathway. So coming to the definition of what is glycolysis. So we can define glycolysis as the sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. Now coming to the salient features of glycolysis. We all know that glycolysis, it takes place in all the cells of the body and it mainly occurs in the cytoplasmic fraction of the cells. And glycolysis takes place in the absence of oxygen, which we call it as anaerobic process, or it also takes place in the presence of oxygen. If it is taking place in the presence of oxygen, we call it as aerobic. And at the end of this anaerobic conditions, if glycolysis is taking place, what we get is lactate. And if it is aerobic, the end product of glycolysis will be pyruvate. So this pyruvate, it is further oxidized to result in the formation of carbon dioxide and water. And glycolysis, it is a major pathway through which ATP synthesis takes place. And this pathway, it is very, very essential for the brain to meet and produce its energy from glucose. This is the summary of equation if glycolysis is taking place under anaerobic conditions. So we see that glucose is converted to lactate under anaerobic conditions. And one more thing is glycolysis, it is also considered as the central metabolic pathway, as many of the intermediates which are produced in this pathway are useful for the synthesis of acids as well as fats. Reactions of glycolysis. So the entire pathway of glycolysis, it can be divided into three distinct phases, and the total number of steps involved in glycolysis are 10. So these three phases, they are the energy investment phase, which we call it as the primary phase. Next we have the splitting phase, and lastly the energy generation phase. The first phase of glycolysis, we call it as the primary stage or energy investment stage. It involves three series of reactions. The first step is phosphorylation of glucose to form glucose 6-phosphate. This phosphorylation reaction, it is catalyzed by the enzyme hexokinase or even glucokinase. So this is a reversible reaction and it requires ATP as well as magnesium ions. The enzyme hexokinase, it is present in almost all the tissues of the body and it can phosphorylate various hexoses. Apart from glucose, it even phosphorylates fructose and mannose. The enzyme glucokinase, which I was saying, it is present in the liver and it phosphorylates glucose alone. So glucose is phosphorylated by glucokinase to glucose 6-phosphate in the liver. This glucose phosphate, as such, it is impermeable to the cell membrane. That means cell membrane does not allow it to cross over. So this is the major central molecule which is produced here, and it has wide variety of fates. That means it can go ahead and proceed with the glycolysis, or it can go to glycogenesis, or it can enter the gluconeogenesis pathway, the reverse one, or even the pentose phosphate pathway. This glucose 6-phosphate which is formed, it is now isomerized to produce fructose 6-phosphate. And here we have the enzyme phosphoglucoisomerase or phosphohexose isomerase. So this phosphohexose isomerase converts glucose 6-phosphate to fructose 6-phosphate, and the enzyme requires magnesium ions for its activity. Next is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The enzyme which phosphorylates fructose 6-phosphate to 1,6-bisphosphate is phosphofructokinase. ATP is required in this reaction, and this reaction is reversible, and it is mainly the regulatory step of the glycolysis. The phase 2 of glycolysis is the splitting phase. It comprises of both the 4th and the 5th steps of glycolysis. The fructose 1,6-bisphosphate which is formed in the previous step, it's a 6-carbon molecule which has to be split into 2 3-carbon compounds. The 2 3-carbon compounds are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This splitting reaction, it is catalyzed by the enzyme aldolase. This is the 4th step. Now coming to the 5th step. In this step, it is again a reversible conversion between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So here, the reaction is catalyzed by phosphotriose isomerase, which catalyzes the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So whatever DHAP which is produced here, it is converted to glyceraldehyde 3-phosphate. So you see that one molecule of glucose, it is resulting in the formation of 2 3-carbon molecules. Here the 3-carbon molecule is glyceraldehyde 3-phosphate. Next phase is the energy generation phase, which comprises of steps 6 to 10. So whatever glyceraldehyde 3-phosphate is formed in the previous splitting phase is now converted to 1,3-bisphosphoglycerate. This reaction, it is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this step, 2 molecules of NADH plus H plus are produced. This NADH, it enters the electron transport chain, and we all know that each molecule of NADH will result in the formation of 3 ATP molecules. So the end product of 6th step is 1,3-bisphosphoglycerate. Now coming to the 7th step, which involves conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. The enzyme that is involved in this is phosphoglycerate kinase. So here one molecule of ATP is produced as 1,3-bisphosphoglycerate loses its phosphate to ADP, resulting in the formation of 2 ATP molecules. And the product is 3-phosphoglycerate. So this is an example for substrate level formation, substrate level phosphorylation, as ATP is directly synthesized from the substrate without involvement of any electron transport chain. And of course, this is a reversible reaction. Next, 8th step is wherein the 3-phosphoglycerate is converted to 2-phosphoglycerate. Here, the enzyme that is involved is phosphoglycerate mutase. And this is an isomerization reaction, 8th step. Next is the 9th step. In the 9th step, you have 2-phosphoglycerate, which is converted to phosphoenol pyruvate. So here, the 2-phosphoglycerate is converted to this high energy compound, which is phosphoenol pyruvate, in the presence of the enzyme enolase. This enolase enzyme, it requires magnesium ions or manganese ions for its activity. And one more thing is, this reaction is inhibited by fluoride ions. So now we have 2 molecules of phosphoenol pyruvate produced from glycerate in the 9th step. Next one is the 10th step. In the 10th step, phosphoenol pyruvate is converted to pyruvate, 2 molecules of pyruvate. So here phosphoenol pyruvate is a high energy compound which now transfers the phosphate group to ADP, resulting in the formation of ATP. This is again another example for substrate level phosphorylation. So we have 2 steps of substrate level phosphorylation. One is here and the other one is here. And this reaction is catalyzed by the enzyme pyruvate kinase. And this is pyruvate formation is the end of glycolysis. This is the last product which is formed of glycolysis. From here, the products will enter citric acid cycle or the TCA cycle. Conversion of pyruvate to lactate. So once the pyruvate is formed, what is its fate? It is the end product of glycolysis. And this formation of pyruvate is totally dependent upon the availability of oxygen. If oxygen is there, then pyruvate is formed. Under endothermic conditions, we have seen that the pyruvate gets converted to again lactate, okay, in the presence of NADH, and the enzyme lactate dehydrogenase does this. So here, the conversion of pyruvate to lactate, NADH is utilized, and we have seen that the reaction could be catalyzed by glyceraldehyde 3-phosphate dehydrogenase, or we call it as lactate dehydrogenase. Now, this lactate, it allows regeneration of formation of lactate, it allows regeneration of NAD+. So since NADH is consumed here, it will release NAD+, which is ready for another cycle of glycolysis. This is same in skeletal muscles, like all this happens in the skeletal muscles during exercise when oxygen supply becomes limited. The amount of ATP that is produced in glycolysis, we'll see that. Under anaerobic conditions, it is just 2 ATP molecules which is produced, while under aerobic conditions, we have around 8 or 6 molecules of ATP, they are produced, they are synthesized from each glucose molecule. Only in glycolysis I am saying. Okay. So coming to the table, we have the pathway, enzyme and number of ATP produced. So we are dealing with glycolysis, here the enzymes and ATP. So at this step wherein glyceraldehyde 3-phosphate dehydrogenase is catalyzing it, we have 2 molecules of NADH which are produced. We know that NADH enters electron transport chain and each molecule of NADH can result in the formation of 3 ATP molecules. So 2 NADH will result in the formation of 6 ATP molecules. So this enzyme at this step, it will lead to synthesis of 6 ATP molecules. Next we have 2 substrate level phosphorylations, right? One which is catalyzed by phosphoglycerate kinase, the other one which is catalyzed by pyruvate kinase. Here at this step, phosphoglycerate kinase, we have two molecules of ATP which are produced, while at the step of pyruvate kinase, we have again two more molecules of ATP which are produced. So you can see that it is a total of 6 plus 2 plus 4. That means 10 ATP are produced at this step. While we also have another reaction, two reactions actually, where ATP is consumed. The first one, which is catalyzed by hexokinase or glucokinase, where glucose is phosphorylated to glucose 6-phosphate. So, one ATP molecule is consumed over here. While in the other step, you have phosphofructokinase, which phosphorylates fructose 6-phosphate to fructose 1,6-bisphosphate. So, here one more ATP is introduced into fructose, that is into the fructose 6-phosphate. So, that means a total of 2 ATP are consumed, hence it is kept -2. So, what we have is 10 ATP produced while 2 ATP are consumed. So, there is a net of 8 molecules of ATP. So, next is we also have pyruvate dehydrogenase step. So, in the pyruvate dehydrogenase step, you have again 2 molecules of NADH which are produced. So, here 2 molecules of NADH means they enter the electron transport chain and they will result in the formation of 6 ATP. So, you have 6 ATP presented over here. While this is the net ATP which is produced in glycolysis under aerobic conditions, the topic that we are dealing. So, you have a total of 8 ATP, net ATP produced in glycolysis. There are 3 irreversible steps of glycolysis which I have been mentioning previously. So, these steps, they are mainly catalyzed by hexokinase, phosphofructokinase and pyruvate kinase. Mainly these 3, they will be regulating glycolysis. Regulation of glycolysis. As mentioned, 3 enzymes will be regulating glycolysis, which are nothing but the hexokinase, phosphofructokinase and pyruvate kinase. Hexokinase is the enzyme which is inhibited by glucose 6-phosphate because hexokinase phosphorylates glucose to glucose 6-phosphate. So, this inhibits hexokinase enzyme. And this enzyme, it will be preventing accumulation of glucose 6-phosphate. Okay, and since the end product of this is regulated, you call it as product inhibition. The product of this enzyme is inhibiting hexokinase, hence you call it as product inhibition. Next, glucokinase, it is the enzyme which phosphorylates glucose. And this enzyme, it is considered to be an inducible enzyme. If glucose is present, its production is induced. Next one is phosphofructokinase. This is one of the most important regulatory enzymes of glycolysis and it catalyzes mainly the rate limiting committed step of glycolysis. Okay, so phosphofructokinase, it results in the formation of fructose 1,6-bisphosphate from fructose 6-phosphate. So next, this enzyme it's also an allosteric enzyme and its activity is regulated by ATP, citrate as well as H+ ions. So these 3 factors, allosteric inhibitors, they will be regulating this. So here we have fructose 2,6-phosphate, ADP, AMP and pi. So that is inorganic phosphate. These are considered to be the allosteric activators, while these 3 are the allosteric inhibitors, that is ATP, citrate and H+ ions. While fructose 2,6-phosphate, AMP, ADP and inorganic phosphate are allosteric activators. So to summarize, we have seen a basic introduction to what is glycolysis, what are the salient features of glycolysis, reactions, all the steps, the 3 phases and the 10 steps we have seen. Then conversion of pyruvate to lactate, production of ATP, what amount of ATP is produced in glycolysis. Then the irreversible steps, the 3 important ones. And finally, regulation of glycolysis we have studied. The books that are used while making this video include The Principles of Biochemistry by Leninger, U. Satyanarayana Biochemistry, as well as Harper's Biochemistry. Thank you.